as fun as you guys might imagine because it's mostly meetings, and meetings with my students, with my collaborators, like different team meetings, writing grants, which is like a really big part of the work, writing papers, reviewing papers, writing letters of recommendation. So a lot at my desk, you know. And then when it was not COVID, at least I had people coming in and out. Now it's just me and my computer. And then I try to be hands-on with my lab and with experiments. We do surgeries for our experiments, and all of those surgeries, I am like the main surgeon, and then other people help me. So the days that we have surgeries, I'm just there fully. Towards the end of my PhD, I was introduced to optogenetics, and optogenetics was basically actually introduced in the neuroscience field in 2005, so it was just relatively new, but it had a lot of advantages, like for example, for optogenetics, because you're controlling the activity of neurons with light, then you can record simultaneously, so you see what's happening in the brain, and you can investigate the direct effects of stimulation, versus with the other ones, like with electrical stimulation, magnetic stimulation. During stimulation, you know, your signal is corrupted, and the other reason was you can actually basically have, because you have cell type specific manipulation, then you can study the role of each specific cell type in the mechanisms, right? You can see, okay, if I only activate these types of cells, then what will happen? Can I, you know, facilitate this rewiring that I want to induce or not? And then the other thing is that, another cool thing is that, for example, you could do multicolor optogenetics. So you can have one color controlling one group of cells. And so opening up a lot of opportunities. And the other thing is that with optogenetics, you also can control to activate or deactivate, right? That's also like another advantage. So the idea of the SmartDora is that I had to develop technologies to enable us to basically study the brain, answer those questions, see what happens when we stimulate the brain, how can we stimulate the brain to rewire it in a targeted way. For example, if there's a stroke, can we use this technology, intervene, rewire around the site of injury to induce functional recovery. You know, it's flexible, so it can sit on the surface of the brain and it can conform to you know, the curvature that you see on the surface of the brain. We started this project last year in collaboration with a colleague, Dr. Yazdan-Shahmorad at Carnegie Mellon University. So they are experts in nanofabrication, and so they're bringing all of that to the table, and we are bringing the design constraints from the in vivo implantation aspect. We have the first version of SmartDora. Yes, we have tested that in non-human primates, and we have tested them both neurally and behaviorally in combination with like optogenetic. This time is an optogenetic deactivation. So we kind of are showing that you can create like temporary lesions. You can think about it. So you're temporarily deactivating different areas of the brain, and then you see during that temporary deactivation how that influences behavior. But this version that we have, the number of electrodes are small, and the number of stimulation locations are also small in comparison to what we want to achieve like in like 4 or 5 years. And then what we have done also is we have developed a model of a stroke that is combined, can be combined with our version 1 of SmartDora. We're calling it multimodal artificial Dora. We can use this photochemical technique and induce an ischemic stroke. And because it's photochemical, we can control the size of it. What you can do is come back and do this imaging of blood flow. And so then we can see that actually now the blood flow has stopped to the area that we create a stroke. And we could do that through our version 1 SmartDora. So there's several advantages there. Now we know where the stroke is, right? We can confirm it in vivo, like, and then we can also then say, okay, now how can we take advantage of like the remaining healthy areas and rewire? And because this is in awake behaving animals, then we can see what is the behavioral deficit that we induce because of the stroke and how can we improve that behavior. Another thing that is also really valuable with this technology is that now we can record from the network. So the best part of working in the field from my perspective is that this is a highly interdisciplinary field. You get to work with people from all different disciplines and backgrounds, like, talks about electrical engineering, mechanical engineering, material science, mathematicians, statisticians, physicians, neurosurgeons, neuroscientists, and I think some of you said bioethics, so philosophers, right? And also, there are some collaborations with the business school as well. So it's a very interdisciplinary field, and I think this is just the beauty of bringing people together and making something possible that's not possible with just one discipline or one individual. I think I just really think that's really, really nice. If I just think about it, I guess one of the things that you guys already alluded to is just the limitation of the technology to be used for, a larger population. I think that's we are so limited. And I think it's just everybody has this vision of how we can basically change the world with these technologies and help people who are suffering from neurological disorders. The numbers are really high. But I guess maybe the way to think about it is that we're at the infancy of this field, and so when it's more mature, hopefully it can be more widely accessible. This part of it is histology, so we do sectioning and staining. There's electronics, if you want to build things, or so this is so you see there's this gray area, right? So that's stained for optogenetics. So the area that's darker has optogenetic expression. So those are the genes were modified to have the opsins. And so then again, if you put this under a microscope, you can see individual cells that are expressing, but with this way you could just see how much expression you had, where was your expression. So that's the different types of staining in comparison to the purple ones that I was showing you before.